Hi everyone, I'm Mani Khanuja and I'm Artificial Intelligence and Machine Learning Specialist Solution Architect at AWS. And today we are going to talk about protein sequence classification using natural language processing techniques. So we will cover what proteins are and why really we need to classify the proteins, and then we'll get into some of the challenges with the existing solutions and what's the intuition behind using natural language processing techniques. Since we'll be using Broadband model for doing the protein classification, we'll understand at a high level what is a Broadband model, followed by the demo of the solution itself. So let's get started. So proteins are macromolecules which are composed of amino acids. There are 20 essential amino acids and each of them is represented by a capital letter. And they together combine to form a protein sequence. Now, why do we really need to classify the proteins, right? So the reason is that the study of the protein localization is really important to comprehend the function of the protein. And it has really great importance for drug design and other applications. And I'll explain that with an example. So if you look at this image, right? So it has a S1 spiky S1 protein of the SARS-CoV-2 virus and the human receptor ACE2. So that really binds to form the pandemic that we are currently in. So in order to disrupt this binding, it's really important to understand the function of the protein and hence the protein localization, which means the location of the protein in a cell, right? So let's understand the challenges that we have with the existing solutions. So currently existing solutions primarily involve doing a database search for evolutionary related proteins. And since the databases are growing exponentially, it's very time consuming. Furthermore, the performance really deteriorates of this solution if the number of related proteins are not found. That calls that there is a need for an alternative solution. So let's see how we can use natural language processing for the protein sequence classification use case. So if we look at the protein sequences, they are made up of amino acids, which are donated by capital letters. So the whole idea is that we consider the protein sequences as sentences and the amino acids as words and use the natural language processing techniques. Moreover, now these protein sequences are really constrained to adopt a particular 3D shape, which we also refer to as a 3D structure. And these constraints mirror the rules of grammar and meaning in natural language. So that's why we can directly map these algorithms onto the protein sequences. Now, since we have understood how we can, the intuition behind the natural language processing, let's look at the Broadband model. So we will use Broadband model, which is a pre-trained model. It's based on the BERT model, and that's where the name derives, BERT. And since it's trained on large corpus of protein sequences, PROT. So PROT, BERT. And it was first introduced in this research paper, the link for which I have mentioned in the slide below. So please check out this research paper as it will give you a lot more details that I'm covering in this demo. Now we understand, now so far what we have done is we talked about what is a protein sequence, right? What is a protein sequence, and why do we really classify those proteins? Challenges with the existing solutions and the intuition behind using natural language processing techniques for doing protein classification. And at a high level, we touched based on the Broadband model. Now let's dive deep into the demo of the solution, which will involve a code walkthrough. In this notebook, and I'm using SageMaker for developing the solution, and we will use this notebook. But before we start the notebook walkthrough, let me cover a few important points. So what exactly we'll be doing in this notebook? We'll download our data of the protein sequences, which is the deep block data set. We'll do some exploratory data analysis on it. Then we'll start with the training, followed by the deployment and prediction, right? So let's first start with downloading the dataset and how the dataset really looks like. These initial cells, we are just setting up the SageMaker environment, downloading some of the, making sure that we have the upgraded and the latest version of the packages. Then we are importing some important packages over here, checking the version of the SageMaker. Nothing much over here, just all the housekeeping stuff. Now, since our data will finally go into an S3 bucket, so that's why we are mentioning the S3 bucket name and the path. Right, now let's look at how the dataset will really look like. Now, we are using the block dataset, which is in the FASTA file format, and it will look like this. It has a protein sequence, and then it has a protein class, which is the location of the protein in a cell, and then it has a label test or train. Now, based on this, we will divide our data set into test and the train format. But before we do that, let's download the data set. So we are just downloading the data set in the cell 6. And this bio package is basically required to read the FASTA file. So we'll read the FASTA file into a pandas data frame. At a high level, when we look at it, we see that it consists of an ID, a protein sequence, a sequence length, and the location of the protein, right? So that's what our data set really consists of. Now let's start with the data exploration. In the data exploration part, we will do 3 things. First, we will understand the number of classes that this data set has, the number of unique classes. Second, we'll look at if it has missing values or not. And then third, we'll see the distribution of the sequence length over the data set to select the maximum length which is required by the model. So that's what we are doing, looking at the null values, and we see that there are no missing values, which is really good. Followed by, okay, let's see the number of unique classes. So we have 10 unique classes. So we basically classify the protein sequences into these 10 classes. Now let's look at the sequence length distribution. So as we can see that most of the data will be actually covered if we select a sequence length close to 1536, right? So if we select the maximum length equal to that, then we can cover most of the data that we have. So this is very clear from our distribution, right? But in this particular sample use case, if we select such a long protein sequence length, it might just increase the training time. So for this sample notebook, we are using the maximum length of 512. So what does that really mean? So when we select the maximum length of 512, then any protein sequence greater than that length will be truncated, and any protein sequence shorter than that length will be padded. And we'll see that when we encode our protein sequences. Over here, we are just factorizing the protein classes. Okay, and then we will divide the data set into the training and the test based on the is-trained column. And then finally, once we have done our exploratory data analysis, we'll upload the data to S3 because that is what is required when we do the training in SageMaker. So what we have done so far, we downloaded our data set, we did some exploratory data analysis, and then finally uploaded the data to S3. And we'll be using 512 as the maximum sequence length. Now let's get into the training script. So since we'll be using PyTorch, so we will write a training script in the PyTorch, and we'll see how. So this is just printing the training script, but let me open the training script in a separate window and explain you that. So in this training script, first we are just importing Torch and other libraries which are required. But most important part is this network definition. So there are 2 things we are doing. One, we are importing the protein classifier class, which is in the model_def file. And the other part is protein sequence dataset class, which is in the data prep file. So let's see what these classes really are. So what I have done in this model definition class is I'm downloading the pre-trained model from the Hugging Face library, and I'm using Broadband BERT localization model, which is trained on a large corpus of protein sequences. And once I download that data, I'm adding another layer, a classifier layer, which will be used for classifying the protein sequences into those 10 unique classes. And that's what we are doing. We are passing the number of classes so that the classification layer produces the output based on the number of classes, right? Nothing much. All we are doing, downloading the pre-trained model and adding a classification layer so that our model can classify the protein sequences. But this model, which since it's based on the BERT model, right? So this model really needs our protein sequences to be encoded. So that calls that we have to write some data preparation function to do so. That's what I'm doing in the protein sequence dataset class. It's very simple. What we are doing over here is we are using the tokenizer from the BERT model, from the Broadband model, and using the encode underscore plus function. We provide a protein sequence for encoding. Truncation true, which means if we select our maximum length, if the maximum length of the protein sequence is greater than what we have selected, in this case we have selected 512, it will truncate the protein sequence, right? And then the padding is also based on the maximum length. So if our protein sequence length is shorter than the maximum length, then it will pad this protein sequences. And that's what it's going to return. It will return the encoded protein sequence with input IDs, attention mask, and targets in the torch tensor format. This encoded protein sequence will then be passed into our model that I just showed you during the training to produce the results. Right? Now let's get back to our training script. And another point that we are doing over here is we are using SageMaker data parallel distributed library. So that's why I'm importing SM distributed data parallel libraries over here. Now again, we are using maximum length is 512, pre-trained model name, tokenizer we are initializing over here, right? So now what exactly we'll be doing in this script? We'll first create a trained data loader because we are using PyTorch and that is what is required. So we'll call our protein sequence dataset, encode the protein sequences, and then use the data loader for the training dataset. Exactly same thing. That's what we are going to do for the test data loader as well. So these are the two helper functions which are going to help us with it. Next, I have also written another helper function which is freeze. Since the Broadband model is really big, so that's why I'm freezing some of the layers so that I can reduce the training time, right? It's a really big model which has close to 31 layers, and I will freeze for the training purpose. I'll freeze the 15 layers, but it's really a choice. You can also try to, freeze less number of layers or don't freeze the layers at all. But what I've seen is that it helps in reducing the training time. So try out with by freezing different number of layers and see how it goes. And the number of frozen layers will be passed as an hyperparameter. So you have the flexibility of changing that. So these were the three helper functions that I've written. Now the training function itself. It's based on native PyTorch code, right? So first we will get the training data using our get train data loader helper function, then the test data using our test data loader helper function that we have created. We load the model, the protein classifier model. We load it. We'll then call the freeze function to freeze some of the layers during the training. This is the distributed data parallel library, because we want to distribute our data into different GPUs that our instance will have. Then optimizer scheduler required for the later steps, so that we can, and we are using cross entropy loss, right? So during the training, what we are going to do, we'll enumerate through our training data set, pass that data set into our model which is in the train mode, and then calculate the loss and iterate through the different steps, right? So that's what we are going to do for the training part. So very basic, standard PyTorch training code, nothing much over here, right? And then once we are done with the training, we'll save our model. And that's what we are doing over here. We are saving our model in the model.pth format, which after the training will be saved into an S3 bucket. So that's what we did in our training function over here. Now let's get back to the notebook and see how we are going to integrate these functions, right? Okay, so I'm scrolling up because I really explained to you what the training.py function has and the steps inside it. So I'll scroll it quickly. Okay. Now we have to do training on SageMaker. So since we are using PyTorch, we will create the PyTorch estimator class. So there are three things that we have to take care of. So when we provide the PyTorch estimator, we need to provide the framework version. So framework version is the PyTorch version that we want to use. The second part is we will use the train.py script that we have mentioned, right? So that will act as our entry point. And the source directory is the location, basically the name of the folder, which will have our train.py script. And you can also specify requirements.txt if you want to download any additional packages into your training instance, right? So that's what we are going to do. And then we are also using SageMaker Data Parallel, which currently supports model training on only these three instances: P316 extra large, P3DN 24 extra large, P3 4D 24 extra large. But in this sample notebook, we will use P316 extra large, which consists of 8 GPUs. And then you also have to mention the distribution strategy. We'll see that in our estimator. Okay. So since we are training on the protein sequences, so our training data set contains close to 11,000 protein sequences. So, and our algorithm is also huge. So this will take about 20 to 25 minutes for the execution. And we'll only iterate through 3 epochs because we want to run the training faster, right? So let's look at the estimator and the points that we recently covered. So we are creating the PyTorch estimator, providing the entry point of this training script. Training script is inside the code folder, which we have. As you can see, it's a code folder that we have, right? And it also has requirements.txt file to download additional packages if needed onto the training instance. Framework version is the version of the PyTorch. This is the Python version. Now we are providing the instance count. So we want to train on one instance of the instance type P316 extra large, which has 8 GPUs. And then we are providing our distribution strategy, which is data parallel, right? Now, once we have provided the parameters for our estimator, the next step is to provide the hyperparameters to this estimator. So we are training on 3 epochs. We are providing the number of classes, which are 10 in our case, the batch size, test batch size, and the frozen layers. And initially in our training script, we said that I mentioned that I will freeze 15 layers. That's what I'm doing, but you can change this number, try with freezing less layers or more layers and see how it goes. So these are hyperparameters, and hyperparameters really are really used for optimizing the algorithm. So you feel free to try it out. And another point to note over here is I've kept the batch size as 4, right? So because if we increase our batch size, then this might give you CUDA out of memory error, right? And our maximum length that we have selected is 500. Maximum protein sequence length that I've selected is 512. So for that maximum sequence length, this batch size works nicely. But if you want to optimize the algorithm further and use a bigger sequence length, then you would have to reduce the batch size. Otherwise, you might get CUDA out of memory error. Okay, so keep that point in mind. And then during our training, these are the metrics that we are capturing. We are capturing training loss, test accuracy, and the test loss. So that's what we are providing it over here. Now let's look some of the results of our training, right? So estimator.fit will trigger our training. In this, inputs underscore train basically contains the S3 location of our training data set, S3 location of our test data set in the inputs underscore test variable, and estimator.fit, it will trigger the training. So let's look at the training itself. It's just downloading some of the requirements packages that I've listed in the requirements.txt file. So scrolling down, providing some details about the job and some environment variables, the hyperparameters, it's capturing all those hyperparameters. Let's look at the accuracy of our first epoch, which is 42%. Then the second epoch, our validation accuracy is 64%. It's improving, which is a good sign. Then the third epoch is 65%. So we have only used 3 epochs because we wanted to limit our training time, but in order to, and we have used the maximum length of 512, so I would say this accuracy is pretty decent. And you can further optimize it by trying bigger sequence length, increasing the number of epochs, and tuning other hyperparameters. And if you need more details on it, please refer to this research paper. It has details on how you can do the optimization and also compares the performance with other models as well. So feel free to try that out and get into the details by reading this research paper. So now what we have done, we are done with the exploratory data analysis. We have trained our model, and after the model is being trained, it will store the model as model.tar.gz into an S3 bucket. So we'll fetch the location, S3 location of the model. So that's what I'm doing over here. And this will be used in the deployment step, right? So exploratory data analysis done, training done, let's deploy the model. Now, in order for the deployment, I have written inference.py script. So what do we need and what does this inference.py script really has? It has 3 functions. First, when we want to do the inference, we first have to load our model. Once the model is loaded, we have to prepare our inference data and encode it because we have protein sequences and the model understands the encoded sequences. So we will encode the protein sequences and then really use those encoded sequences for the prediction. So that's what our inference.py script has. Now, in order to kick off the deployment, we first have to define the PyTorch model object. And over there, we provide, okay, where my model data is, that is the S3 location of my train model, source directory, inside which I have inference.py script with these 3 functions that I mentioned, and role and the framework and the Python version. So now we have this object that we have created, which provides the necessary inputs. Then we'll just do model.deploy. Now when we are deploying the model, we have to mention the instance on how many instances we want the model to be deployed. It's a sample, so we are deploying it on 1 instance. Then the instance type. We are using a smaller instance, and if you look at it, it's ML M5 to extra large, which is a CPU instance, because we are doing inference. We are not training, so not a bigger instance is required. So this is pretty decent for all the prediction work that we'll be doing and provides it with nice latency. And if you want to further improve the performance latency, please try out elastic inference that we have, and that can further improve the performance. And then the endpoint name. So when we do model.deploy, what it is going to do, it will deploy our trained model, which is on this S3 location that we mentioned in our PyTorch model object. Use this inference.py script to load the model, to convert the protein sequences, encode the protein sequences, and do the prediction, right? So now we have deployed our model. Now, once the model is deployed, let's do some prediction and see how our model is performing. So before that, we are just describing our endpoint, provides you with good metadata information, right? So just for the information purpose, our input will be our JSON input. So our protein sequence will be in a JSON input format. That's why we are using the JSON serializer and deserializer. Now I'll provide a test protein sequence. We'll do predictor.predict to predict the protein class. And if you look at it, the ground truth is endoplasmic reticulum, and that's what our model has also predicted. Similarly, I'm trying it for other protein sequences as well. And so far I can see it's giving me good results. That's what I wanted. But for you, it might give you different results, because remember, our accuracy is only 65%. That's why, but feel free to try it out and provide us the feedback on it. And once you are done with the predictions, because it's a sample, and if you don't need your endpoint any longer, please delete the SageMaker endpoint, as it will incur the charges. And that's what I have added the code for, right? So that was all about the demo. In this demo, what we did, we downloaded the protein sequence data, which was in the FASTA file format. We then did some data exploratory analysis, looked at the number of unique classes, decided our maximum sequence length, and then split the data into the train and the test, uploaded the data on S3 bucket, did the training using the train.py script, did a whole walkthrough of that script, and then after the training was done, we talked about a little bit at a high level on how you can further optimize the algorithm, and then we deployed our model, and followed by doing some prediction on the test data. And I've also added the references in the notebook. Additionally, I have also provided the references in the presentation. So in this presentation, the code is in the GitHub repository, which I have mentioned. Feel free to check that out. Clone the GitHub repository, try it out on your own, and provide us feedback on it. We are always happy to incorporate the feedback. And in this case, I was using the Hugging Face library. The pre-trained model was coming from the Hugging Face library, so I provided the model link for that. And now we also have, SageMaker also supports Hugging Face library inherently, so I provided the links for that also, which might be useful. We have link for dataset, research paper, PyTorch SageMaker, SDK, and the distributed data parallel that I used in the code. So feel free to check it out. And these are some of the nice resources, and provide us your feedback on it. Thanks for tuning in.